I was going to start by just kind of showing off what is the Xylariaceae, and I think we will go back to that later. Because I actually would like to start with just the leaderboards. This is kind of we do it every time. Let's look at who's ahead, and then we'll get into looking at some actual observations. So right now, this is our 2024 project. We are about 12 days away from the end. We have right here at the end 102 members, probably the biggest ever. And of those, the people who are leading our leaderboard for most observations is Tanner, just recently passed Matt. And then we have Liz with mushroom photography, Hedgehog, Brennan, and Jeremy Hooper as sloth power. And so those are the rankings. I'm not on them. I, like a lot of people, have more observations in my photo roll than are up here, and I'll have to spend some time adding them. So just a reminder that whenever you add them, if you've joined in the project, even if you add them February, they'll add into this project. So there's still time for you to get on the leaderboard for either most observations or most species. This is tabulated by how many all the way to species research grade observations do you have. And so we have Matt again, Liz, this is Tavis, Hedgehog, Tanner, and Brennan with most species. And it's just kind of a point of curiosity to see, well, what are the most observed species by our members? We have pheasantback, crown-tip coral, turkey tail, what do we call this one? I'm blanking. I call it Ishno a lot of the time. Salisbury snake of the woods. So we have some background audio there.
Glenn, you did mute.
I have muted him. And I muted some other people who weren't muted, but if anyone wants to unmute themselves to talk, please feel free to do so. So now getting through the leaderboard, we can actually look at some cool observations. So as I said, I was gonna this is what Fenner is gonna be talking about for us today, the family Xylariaceae. And I was just kind of curious, first of all, what in this family have we seen in our projects? So to do that, I went to our all WMS projects, umbrella project, and then you can click through to observations. And when I do that, you can search for a given any level of taxonomy. So we could search and click on family Xylariaceae. And when we do this, this is what that is. I kind of preloaded everything so it didn't spend time. So this is what people are seeing in our WMS projects in that family. And you can also look at the map to get a sense of where are people seeing this. So people in our WMS projects have been to the United Kingdom, they've been to Guyana, they've been somewhere in the South Pacific, probably Hawaii. Yep. They've been to Venezuela, Suriname, all over the United States, but of course most are in Wisconsin. And this explains some of our, observations that I wouldn't more expect to be in Wisconsin. We could check this one right here. A lot of Xylaria are, as I'm sure Fenner will get into, super host specific. This one is on a magnolia cone, and it's named after
You see that a lot down south.
Hunter saw this one in Louisiana.
There's also Xylaria liquid ambar, which is named after, I think that's a species name also for a sweet gum tree, and it grows in the sweet gum cones similarly.
Oh yeah, you're right. Very cool.
So I don't know, it's hard to imagine what's so unique about another piece of forest detritus, but it's really interesting that, those two species have carved out, just like I know exactly what to do with this exact detritus. All you other microorganisms can buzz off.
Yeah, they're right there, ready to colonize the moment they can get into it, is what I've heard, probably from Fenner actually. But going back to what we've all seen, so you can, if you're looking at, say what everybody in WMS has seen in Xylariaceae, you can also then look by species. One of the things, a lot of these surprised me until I remembered that some of our members have been to places like Guyana, like this is from there. But beyond that, something surprised me because most of these are Xylaria. We sometimes forget that brittle cinder or our Kretschmeria dusta is also in it, but also things like these Peronia species, which this is kind of hard to see, but I definitely understand that. If we click through to the genus, we can see some of like the top pictures in this genus. And this is probably kind of a better representative. And of course, sometimes people put up the microscopy. It's very beautiful.
We love to see microscopy.
We love to see microscopy, and it does take quite a bit of work. But it's pretty accessible if you take the pictures through your phone. Nail fungus. Wonder why it's called that.
Yeah, I'd actually heard of this one. Some of these pictures aren't the best. Maybe if you tried Google quick, it
Well,
really
let's
is
see if
kind
I
of.
pull up. Oh, there. That's a good one. Oh, it looks like a nail, like you hit the nail on the head kind of nail, kind of, or a golf tee, kind of. Weird. And you can see the family resemblance, the Xylariaceae kind of resemblance, especially on the side there, where you've got like the stroma being black
Yeah,
and crusty. But going back to things people have seen in our projects, this is Liz. And I brought this one up. I actually don't know if Fenner's here yet or not, but Michael is, and other people interested in Xylaria perhaps. So Liz had seen this one, and she had put it down as fairy sparkler, Xylaria tentaculata, and she said, or Xylaria hypoxylon. So just because there was some question about this one, even though, oh, wait, never mind, Fenner. I hadn't even noticed this earlier when I pulled it up. This is Fenner on iNaturalist, and so I guess he does agree that it's the fairy sparklers instead of
oh,
Xylaria
oh, what do you
hypoxylon.
know?
What do you know?
Funny coincidence.
Yeah, and this is actually probably one of the cooler Xylaria species that we can see in our region. We can take a quick look in the map right there. And while it's, I think, more common being observed further to the east, Liz found it. I don't know exactly where she was actually. More like Illinois. Like, why do they hate us? Why did the fairy sparklers avoid us? We all need to go out there and find them because they're here. So if we look at the seasonality, it looks like we probably would go out more in August to look for these. I don't know if they have faux specificity. Let's see what we see. Okay, there's some oak leaves in the background.
This is such a good candidate species for showing off just how alien Xylariaceae can be.
Yeah, they're kind of reminding me of those sticky hands that you get at like carnivals and things. Yeah. But I could definitely see the sparkler. So this looks like it's coming out of what, the downed wood here. Now that's pretty crazy. I would like to see that sequenced to compare. Would this person see this? Because that's not just got the branching at the top that has the branching going
Fenner has
down.
joined the chat.
It looks like a baby bottle.
It does. Oh, good. Hello, Fenner. We're just looking at some Xylaria. I don't know if you were here for the, this is somebody in our projects who saw it. When we look at the map for this species. It unusually, not unusually, but there's a gap that we in the Wisconsin Mycological Society could go out now that we've seen it and very specifically be like, they're here, they're probably here. They seem to be much more commonly there, but there's some credible, that is a
Yeah, it's hard to
credible
imagine
observation.
something being in Michigan and Minnesota, but not Wisconsin.
But not us. Yeah, we
I
need
can't
to get
fathom
on
it.
this. We need to get on this, people.
That's the goal this year.
Yeah, bring me 15 tentaculus. Yeah, this looks very credible. All over a shady north-facing slope on the bluff. It's not very far from me. Wow.
Such a fun
I
species
would look
to
at that
find.
and I would think it was just pine needles or something. Like now that I've seen that, I'm gonna have to look and go, oh, that might not just be pine needles.
Yeah, they're
Yeah.
really small too. So usually when I find them, it's like something bigger that brought me there, and then I find Xylaria tentaculata.
It looks kind of like those air plants.
It does.
That's
It
cool. Yeah,
does.
that's pretty accurate.
I want to see one forming too. I think this would be a great time-lapse candidate if you find one.
Do you think those long ones are like it had a big window and it's kind of ideal conditions, and maybe those shorter sparkler looker ones was like stunted?
I'm not sure.
Yeah, we were looking at
think
this in
yeah.
Colorado, and of course there's so much
Whoa.
like hidden diversity, like not yet recorded.
Yeah, I'm not sure I would call that tentaculata. It looks a little different.
It's also in Colorado, which is outside of
Yeah.
its normal. It looks quite different.
No
But-
kidding. I would like to like get a collection of that sometime. That's pretty neat.
Yeah, well, see, that's I remember about it. Yeah, going back to this species, we were just kind of browsing it. Do you know if this one has host specificity?
No, it really just likes super, super, super well decayed wood or even like nutrient dense soil. It'll even grow from like leaf litter and stuff sometimes.
Yeah, it looks like people are
One of
showing
the
it.
only Xylarias I've found on conifer as well. This tentaculata really doesn't seem to care. Hey, there's mine.
Which one?
Up one more. Yeah, that's it.
Dave,
You'll see that picture pretty soon.
Oh,
you're not
cool.
speaking. Could you mute anybody?
Oh yeah, with background noise.
Speaking of which, can you guys hear me okay?
Yeah,
Yeah, you sound great.
Cool.
you sound great. Also, are you doing well, Fenner? Or we heard you were under the weather.
I'll be perfectly transparent with you. I'm not doing well.
Okay, I'm
But
sorry.
as of right now, I took 800 milligrams of ibuprofen like an hour and a half ago, and I'm feeling so good right now. Like it's better than I have
That's
in
great.
like a whole 2 days.
That is great.
Yeah, RSV and COVID at the same time is like crazy.
Yeah, I can only imagine. I don't know if I would be able to do public speaking during that. So
It's tricky, but I love talking about Xylaria, and I don't want to let you guys down, so I'm gonna just do the best I can.
thank you.
Yeah.
We're so glad you're here, and we hope that the healing powers of talking about Xylariaceae things
Yeah.
will
No
be there
kidding.
for you. This was one that I wanted to highlight. This is something that there's only 3 of us in our projects at least that have observed it.
Yeah.
Jack got a really good picture of it in and around La Crosse, and it kind of breaks even though we can see the family resemblance again, it's not our usual Xylaria. It's
Right. It's like Xylaria is like multiple parathecia embedded in a stroma, and then Rosalinea is multiple parathecia embedded in a subiculum. So it's like they're really similar, but like not so similar that you would ever confuse them as one another.
and this
I'm not familiar with those words, stroma and spiculum.
Okay. Yeah. We'll talk about that on the presentation.
Fun.
we
Good.
will.
Yeah.
It's all coming back to me because I learned the word subiculum when I was looking up things about this. and
Yeah.
it's the yellow stuff in the background in this case.
Mhm.
It's
Yeah,
like
Rosalinia, the genus Rosalinia is pretty well known for having a subiculum. And Rosalinia subiculata is pretty unique that it has a yellow subiculum. It's like
Yeah.
the only one that does. So
they
pretty
named it
easy
right
to read.
after the word
Yeah.
because everyone was like, great, perfect. We have a way to identify this.
Yep.
I wonder if anybody's posted microscopy because Xylaria have pretty cool looking spores.
They do. Yeah. Super melanized and we've got the germination slits. That's pretty common across the whole family, even the order.
Mm-hmm.
I was wondering if maybe you could during this time, and I don't know if you're going to get to it in your talk about Cosmospora arxii.
Cosmospora
And
arxii? Yeah.
yeah,
I can talk a
could
little bit about that. I can probably pull up some pictures of it too. I've got a few observations of it.
so that's if I'm remembering correctly, that was a parasite on hypoxylon.
Yep.
Oh, this
There's mine.
red stuff.
Yeah, it's these red parathecia. They're a nectrioid fungus, so they look sort of like a
Oh.
nectria. There's one that grows on Kretschmeria as well. It's
That's what that is.
Cosmospora eustaline. Grows on Kretschmeria, and then
Let's grab
Cosmospora
that up.
arxii. I find it on Hypoxylon howianum mostly. It can occur on other hypoxylons though.
Yeah, the one we saw in West Virginia, or I saw when I was with you, was
Yeah.
Fragiforme.
Yeah.
Down the top, twist them.
It'll grow on Fragiforme. I've only found it on Fragiforme and howianum, but that's a super cool genus too. I'm thinking if I ever get like bored with Xylariaceae, which I doubt I will, I'll probably look into Nectriaceae a little bit because they're super interesting. I especially like the parasitic ones.
It's a very important one if you could figure out how to stop the beach blight,
Yeah, no
Mm.
doubt. The nectrioid fungi are like 9 times out of 10 up to no good whatsoever.
I wanted to just kind of do a side-by-side
An
comparison
absolutely diabolical genus.
to orient people that it's kind of cool that so the parasite that we're talking about in this family Nectriaceae, this Cosmospora, it's in the Cervidiomyces class, and the stuff it's parasitizing, wow, also in the Cervidiomyces class.
Yeah, it's just parathecia on parathecia.
Yeah.
Turtles all the way down.
That's right.
Wow. So I'm just looking at anything in the genus because I was looking for. Yeah, so if you are looking at something that you think is a Xylaria, but you see some weird little red orange stuff on it, start here with this genus, Cosmospora.
Just a real
There's a
quick
few-
5-minute warning that we're going to do a little takeover for about 10 minutes to talk about the elections.
Okay.
So
Well, I'm going to stick in the chat really quick. We are, let's see, 12 days, 5 hours, 14 minutes, 10 counting down seconds away from the beginning of 2025. So I have made this new project for us, for anyone who wants it. I'm going to stop sharing real quick so that I can put it in the chat. So if you want to get started joining the project early and being set to go for the, competing for the first WMS specimen of 2025, you can get in on that. It doesn't yet have the art that will be on the project, but it's there. And I guess for my remainder of my final minutes, unless anyone wanted to bring up.
I was hoping we could just look at any Xylariaceae in the 2024 group.
Yeah, that's a good idea for within the 2024 group. I was looking at ones within our this umbrella project.
That's what I meant.
But we could go just to 2024 and see what Xylaria things did people see. Wow, we almost at 11,000 observations together. That is some good work.
Yeah, nice going, guys.
So let's check out this family within our project. Oh no.
Yeah,
that's
what's the
not what I wanted.
Bluegrass Club putting up? Gotta get those numbers up.
Yeah.
I would say it's not a competition, but it kind of is.
That's fair.
Yeah. So we have a lot of Xylaria polymorpha with one.
That
Oh.
kind of makes sense.
Yeah. This is not one that, let's see, and it
Xylariaceae
doesn't,
corticium.
it's not been confirmed, so we could look at it and see, do we think it's a,
I'll be honest with you, I don't think it is. Yeah, that looks more like bug frass or something.
yeah, let's look at what, yeah, because it's got to have this, or like,
Yeah.
it looks like it has more of this
Corticium's
structure.
got like this wiry, brownish, reddish subiculum.
And so right now I have quality grade any. I can restrict that to research grade so that I can only be looking at things where at least 2 people decided.
Yeah.
Let's see. Yeah, there's more structure there.
Yeah. Yeah, that looks a lot more like the Rosalinea corticium that I'm familiar with.
Which it happens to be sure. That, yeah, it does kind of look, and then there's a cool mold on it.
Yeah.
So this is where you can politely leave a comment like, might be bug frass. Check out that mold though. Wow, I can't type. And that's how you do iNaturalist. Going back to, here's somebody talking about rose.
Cornudame.
Yeah, actually, this is mine. I didn't want to
Okay.
hog all the time, but I'll hog the last couple seconds. This is the longest Xylaria I've ever found. This is 8 inches long.
Yeah.
I found it under this like hollow
Yep.
tree. Let's see how I can, yeah, see.
Oh yeah.
Longest stroma. And it's longer than the description, the type description of the description
Oh,
in,
yeah.
in Rogers, not me Rogers, but a different guy Rogers.
Jack
In his
Rogers.
Jack Rogers, in his
Rest in
description
peace.
of this, which is he
Yeah.
published the only like monographs for Xylaria in North America
He did
that
a
I
lot of great
found.
work. Yeah.
And this is longer than the range. It's still like there's no other
Yeah.
species that it could be, and
Dude, it's such a variable species, but everything about that screams Cornudame to me. Yeah, like it's growing like from buried wood, but like really well decayed. It's got the conidial coat that's retained even at sexual maturity. Super long rooting stipe. Every single thing about that's definitely Cornudame. Yeah.
Yeah, this is the powdery white stuff is that conidial stuff, and these are irregular, kind of crunched up though.
Oh yeah, there's the Parathecia. Yeah.
These are my spores. And this is, I thought this was the germ slit and that it was just, but
It's
I
kinda
also
hard to tell,
knew.
yeah. Cornudame has a straight germ slit. I think it runs like full length or 2/3.
Yeah.
Okay, y'all, I'm crashing your party.
No.
Oh,
Okay.
yeah.
Well, we'll
I
get back to
know.
Xylaria soon.
Yeah. Okay, so just some quick announcements. This is not just a lecture night for us, but this is also our Zoom annual meeting. So we are having an election that opened in November and it is closing tonight for the board. So if anyone hasn't voted and you still want to vote, I can send a link to people. I'm just going to copy the link and I'm going to actually put it right in the Zoom. So you can only vote once. If you've already voted, that's it. That's all you get. If anyone else wants to nominate anyone to be on the board, you can still do that until the election closes, which is just going to be in a couple of seconds here. And I'm not sure you're probably going to get elected at this point because we've had hundreds of people vote. But I'm going to share my screen at this point, and we're going to go over the election results. So we had 124 responses to our survey, and we had 15 open slots and 15 people So all we needed as a percentage to get elected was at least 50% of the vote, a majority of the vote, 51%, I guess, or 50.1. But everyone on the list got elected, and my apologies to the new folks because you missed about 10 votes because I didn't have you on the list right away. But we got them on there, and now everyone is elected. So welcome to the new board. Really appreciate every single person that actually took the time to vote because of the fact that we do need quorum, which is like a certain percentage of the people that are our members to actually like sign in and vote and make it binding for us. And we did get enough people to do that. So thank you everyone for taking the time to do that. Now I'm just gonna take a minute and we're gonna kind of introduce everyone that's on the board so that people can kind of wave hello. We can just say, you know, our names, how long we've been on the board, where we're from, and what we're probably looking forward to for 2025. So I would like to start with, well, I'm talking myself. So I'm Melissa Klotka. I'm the current president, but I'll be stepping down from presidency from this next year. And I have been on the board, I think, since like 2019 or something like that. And I'm from Waukesha, Wisconsin, and I am looking forward to rustic camping the most this year again. It was really fun this last year. So we'll just go in order then if people can. Tavis, you're next on the list. Are you here?
I am. I'm Tavis Lynch. I'm from the northwest corner of the state. I've been on the board for I don't know 4 or 5 years, I suppose. Been in the WMS since the mid 1990s.
What you looking forward
And-
to, Tavis?
Maybe some melting.
What did you say? You broke up. I didn't hear you.
It's colder than hell up here. I want the snow and the ice to go away. We got 12 inches of ice on the lakes, and there's 2 feet of snow on the ground. I'm ready for
Get
it to
out there and
melt.
do some ice fishing.
It's too cold.
All
Well,
right. I don't know if Arnie is here or not. Is Arnie Martinson a part of the crew right now?
I think he's in
Can
Europe.
someone check? No?
Yeah. I don't think
Okay. Go ahead, Mariah. You're next.
All right. Well, following on Tavis's weather report, I am Mariah Rogers. I've been on the board also for like 5 or so years. I have a hard time with time sometimes, but it was like before the pandemic, and that was about when I joined the board, and then the previous year was when I joined WMS. So it's been a great journey. I'm very thankful for everybody who I've met in WMS, and I'm looking forward to seeing all of you. I'm thinking my plan is still to run for the presidency of the board in this next year. Right now I'm secretary, and Brennan has been steadily but surely taking on literally everything that I've done as secretary. So I have more confidence in him than me at this point on that. And yeah, I'm very thankful to everyone on the board too. Besides looking forward to just seeing everybody, I'm also super excited about some of our potential sequencing efforts and helping out with that at University of Wisconsin La Crosse. So
Cool. Brennan, you're next.
Yeah, that's me. I am a board member at large. I am the assistant secretary. I am the chair of the science committee and some other stuff. I'm a 4A leader out there sometimes. I've been on the board for 1 year, just a little over 1 year. And I am definitely looking forward to getting some DNA sequencing results in 2025 and more iNaturalist observations, including more Xylariaceae and everything else.
Great. Okay, so is Brad here by any chance, or Steph? I didn't see either of them.
I think Emily May was here. If you're going down the list.
I am. I did see a Jeremy join. Maybe Jeremy Homestead's here.
I'm here. Yeah, I'll speak
Great.
up. Yeah, so I'm Jeremy Homestead. I'm going into my third year on the WMS board, and I've been a member of WMS. Oh, I can't remember exactly when I joined. I want to say 2010, 2012, something like that. It's been a while. So yeah, I enjoy the club and I enjoy what we do. So yeah, everybody's cool.
Amen. Okay. Well, great. I know Brooke is not here for sure because he had to fill in for someone on a NAMA webinar tonight because they got really sick as well. So he is doing his NAMA thing tonight. But we're glad to have him. And then Emily May.
Hey, yeah, my name's Emily May King. I'm in northern Wisconsin at a place called Minocqua, and this will be me entering into my second year at WMS, and I'm so honored to be part of this beautiful, beautiful team, and I'm really looking forward to getting out there and mushroom hunting season, and especially interested in starting up the conservation and storage committee.
And you're doing a great job with that so far. So thank you. And Shelby, are you here?
I see
thought I saw
Shelby
her
in the
name.
list, but
I'm here, but
there you go.
I'm using my phone and I don't know how to get the camera to work on my phone either, apparently. So hi, I'm Shelby. I'm going into my second year as a WMS board member. What were the other questions? I'm sorry.
What are you looking forward to and where are you from?
Oh, I'm in Eau Claire, and what am I looking forward to? 2025 as a whole. This year was great.
Good.
Next year will be better.
Excellent. Thank you, Shelby. Walker, you're next.
Hi, I'm Walker Rugeley. I've been on the board. Oh, I'm definitely gonna need that person muted. It's the feedback.
I gotcha.
Yeah, been on the board about 6 months now. Melissa kind of roped me into this. I helped out with the National Forest Service permit. And anyway, it seems like a great way to contribute to a club that I enjoy participating in so much. So I'm looking forward to doing more of that and finding a lot more mushrooms in 2025. So it's good. I'm in Green Bay, by the way. Yep.
Excellent. Thank you, Walker. Okay, Marissa, you're next.
Hi, everybody. My name's Marissa Dorn. I actually live in Chicago, but I spend a lot of time in Wisconsin and I spend a lot of time traveling all over the country. I spent the last 13 years on the West Coast, just FYI for my mycological background info. So I'm really enjoying getting familiar with the Midwest mycodiversity. And in 2025, I'm really excited to be on the WMS board and simultaneously continue learning a ton while also hopefully being helpful. I have a big background in events, so I'm excited to be able to help with some of that stuff. And then I'm also a student of GIS right now, geospatial technology. So I'm hoping that some of that can be helpful to WMS and I can continue learning, especially on the science side of things. So that's me.
Excellent. Thank you, Marissa. And our next new board member, Max Reinhardt.
Hi, I'm Max Reinhardt. I've been part of WMS for a couple, 2-3 years now. From the West Coast. I was born in California. I lived in Oregon for a long time. I'm really excited to see what Marissa Doran brings to the table with the mapping components. It seems very fascinating, and as well as events. I am very excited to participate in whatever way I can on the couple different committees, events, and and things, and finding as many mushrooms as I can and contributing in whatever way.
Excellent. Thanks, Max. And last but not least, our new member, Luke.
Hi, I'm Luke Moat. I've been kind of involved through WMS for a couple of years, mainly because I know Steph Lee and she's brought me along to a lot, but I'm from northern Wisconsin. I'm really excited to be a part of this, mainly because my whole background is in genetics and sequencing and bioinformatics work regarding that. And I'm really excited to maybe use some of those skills, bring them here, help out where I can. For 2025, I'm just excited to see where you guys are at and how you run stuff. And I think that fungal genetics is really untapped compared to a lot of the animal genetics and human disease components that I work in. So it seems like there's a lot of cool work that could be done kind of quickly or easily, I guess. So I don't know, just happy to be here.
Absolutely.
It's also a
Well,
talented guitarist.
Oh, well, that's fun for events coming up. We'll have him and Marissa playing together, banjo and guitar. That'd be great. So I'm gonna introduce our speaker in just a second here, but I just wanted to take a one moment on our very last Zoom meeting with me being the president of the organization to just say thank you to every single person that is on the board. These people that we listed, plus other people that were on the board even just briefly this year. Jennifer Mickey, Peyton Martinson, Sammy Peterson, and Jessica Anderson. Even though you're not going to be part of the board for 2025, thank you for your contributions. Obviously, we couldn't have gotten through 2024 the same way without you, but for the board that we're keeping and the board that we're moving forward with, also thank you for joining us and for everything that you've contributed and everything that we're going to do together for this next year. I think that we have an awesome team and I'm really looking forward to it. And I just wanted to say that we have come a long, long way in the 2 years that that I was president, and I'm just really proud of all of us. So thanks, guys. Okay, well, with that, I'm going to stop my share and I'm going to introduce our speaker. So Fenner Martin Morris is a part-time biology undergrad, full-time worker and director of taxonomy at the Bluegrass Mycological Society. He's an amateur mycologist of 4 years, having dedicated 2 of those years primarily to the identification and taxonomy of the genus Xylaria and its allies. This lecture will serve as an intro to the genus, as well as highlighting the untapped biodiversity found in the eastern United States. The family includes over 1,000 species, 33 genera, with more than half of those species belonging to the Xylaria genus. So without further ado, Fenner, if you want to go ahead and share your screen, you can take it away.
Alrighty. Awesome, guys. You guys see everything all right? Yeah,
Looks good.
see my screen and stuff? All right.
Yep.
So bear with me, like I mentioned earlier, got RSV and COVID, so I'm rocking with a little bit of brain fog. I might have to mute my mic to cough here and there, but this will be a fun presentation. So this is Xylaria diversity in eastern United States. It's a little introduction and notes on, I think, 21 or 22 species. I added a new slide in there today even. So the genus Xylaria, it's characterized as stromatic ascomycetes. They're upright, and stroma in this sense, it means something totally different from botany. A stroma in mycology is it's a type of ascomycete. So I'm sure most of you, if not all of you, know the difference between basidiomycetes and ascomycetes, but Xylaria are ascomycetes, and they're not the cup fungi kind of ascomycetes. They're the flask fungi, so they have parathecia, and it's upright stromata, And the stromata has a carbonaceous outer coating. So it's like the classic blackened exterior that you see in Xylaria. And they're pretty well known for just being super variable with their macromorphology. So microscopy is generally the best way to identify them, because even within a single species, the macromorphology, it can get a little wild. So a lot of these ones that I cover, like the species that I cover, I don't provide microscopic features for, but like in order to identify them in the first place and get familiar with them to identify them, I had to do microscopy like multiple times. So the microscopic characteristics of Xylaria that are important and considered taxonomically stable within a single species are these highly melanized ascospores, like you see right here. They're super dark, usually like blackish, sometimes kind of chestnutty or walnut brown, but usually very black. And the germination slit right here, the ascospores with germination slit. So you see right here, this is Xylaria finismundoensis, by the way. It's a super small Xylaria species. But where you'll find these cells is in the perithecia right here, these black globose structures. And you see here's an ascus, and then there's the ascospore. Got that pale line running right there is the germination slit. It's a straight germination slit. This one runs the full length of the spore. And also really important is when mounting Xylaria sections in iodine solution, it causes a amyloid reaction in the ascus plug or ascus apical apparatus, if you want to be fancy with it. And that's this bluing right here. It kind of looks like an inverted hat, or like two rectangles right next to each other, kind of flares out of the top there. That's a really important feature for studying Xylaria. So if you study Xylaria microscopically, you really need to have an iodine solution like Lugol's or Melzer's or Barol's. So Xylaria is the type genus for the family Xylariaceae, and it contains a couple of oddballs like Rosalinia right here. This is Rosalinia corticium. So in the order Xylariales and the family Xylariaceae. So like we were mentioning earlier, it's very similar to Xylaria, but instead of multiple perithecia embedded in a stroma, this is just these loose perithecia embedded in this wiry subiculum. So phylogenetically speaking, they're super close. You could almost call it a Xylaria, but like macromorphology is unique enough to be a valid genus for sure. The order Xylariales contains stuff like diatripaceae, like diatripe, and like hypoxylaceae, so stuff like hypoxylon, daldinia, all of the other hypoxyloid genera. There's so many. So at the time of this presentation, there's currently 884 valid species of Xylaria per index fungorum. So like a lot of those are probably redescriptions, and but still that's huge. Like that is colossal. And combine that with there only being 23 ITS type sequences for type specimens of Xylaria. There's only 23 ITS sequences, and they're all like newer species that were described post-sequencing era. So none of those are type sequences from classical taxonomy. So you can already see there's a really big issue with reference data. So if you sequence a Xylaria, you're probably going to get like a temporary code name. Nobody has ever sequenced it before. You'll have to do the microscopy anyway, so you might as well just buckle down and do it. And to make matters worse, phylogenetically, a recent paper by Mark Stadler and a few other co-authors talks about the issues with using ITS for Xylaria, Xylariaceae, and Xylariales. They're really known for having what's called intragenomic polymorphisms, which basically just means like incredible genetic diversity within a single species. So a lot of the times, ITS doesn't work super well alone. However, if you use these primers right here, LSU, RPV2, TUB2, so beta tubulin, and ACT1, these are gene regions that code for protein synthesis. It's like super important that they don't mutate and become non-functional. It turns out really good for species delimitation within the order Xylariaceae and all the way down. So something really important about Xylaria is they are not really like a regular mushroom, how they just kind of pop up, drop their spores, and then they're gone in a couple days. They actually last months. They're super long-lived, and they start out as what's called anamorphs, which is basically just asexual fruiting bodies. Anamorphic stromata are covered in conidia, and those are basically asexual spores. Although I will say the function of these asexual spores in the genus Xylaria doesn't seem to be similar to the function of asexual spores in any other anamorphic ascomycetes. They just don't really germinate on agar plates. They don't germinate readily for anything like that. There's been a couple papers about their function and hypothesizing that it might be like it has to land on like a flower of a particular host and stuff. So I don't really have the answer to that. I'm not sure anybody does, but just for the sake of making this digestible, we're going to call them asexual spores because that's what conidia are in any other anamorphic ascomycete. Sorry for the rant. I just had to make that very clear. So the conidia are born externally, and they appear as a very like thick, dusty layer, and common pigments include white, cinereous, which is sort of gray, smoky. They can be bluish, they can be salmon pink, and they can be like off-white tan. So anamorphic material is less desirable for collection and study because there's not a lot of defining characteristics with them. There's not a lot of taxonomically stable traits and cells that you can look at to really pin down an ID. So a lot of people love to send me Xylaria collections, and I love that, but a lot of the time when I get them, they're just anamorphs, and there's not really a whole lot I can do with them. So that's why it's pretty important to know the difference. Once they mature, though, well, first of all, the anamorphs, you can find them in spring to early summer. So once they mature, though, they develop into what's called teleomorphs. And those are like the pretty classic, like blackened stromata with the carbonaceous outer layer that it protects the embedded parathecia within the stroma. And the parathecia are where the sexually reproductive cells are born, such as ascospores and asci. And you can find these in late summer and all throughout the fall into early winter. And sometimes you can even find like last year's teleomorph growing next to this year's anamorph. And a lot of the times that'll be like just like a hollow, dead stroma, but it's better than just having an anamorph. So places you can find them, most commonly found on dead hardwood. I don't really find hardly any Xylarias on conifer. Like I found that Xylaria tentaculata one time, and it's just like an opportunistic one. So you'll find a lot more diversity on hardwood forests. You'll find a whole group of them on fallen fruits and seeds as well. A fructiculus Xylarias. We'll cover a handful of those as well. They grow on leaf litter and petioles as well. Occasionally, they can even be parasitic on living trees. And really, really often, they're living inside healthy plant material as an endophyte. Xylarias are ubiquitous endophytes. If you walked around just about any, even conifers, they're endophytes in conifers and everything. They live inside the plant tissue. So if you cultured like all sorts of material from leaves one day, eventually you would absolutely culture a Xylaria. They are ubiquitous endophytes. They're less common on grasses, sedges, and herbivore dung, although the literature mentions this a multitude of times. So if you do happen to find any on grasses, sedges, and herbivore dung, please reach out to me because I'd really love to study that material. All right, so into the species that we're going to cover. I felt kind of obligated to start with this one because this poor taxon gets overused so much. So many species go misidentified as Xylaria polymorpha, also known as dead man's fingers, which I'm sure many of you have heard. So this one, the literature mentions that it grows in what's called grotesque configurations, which I really like that description because it's just super cesspitous. It has no rhyme or reason. It just grows, and it's grotesque. They're grotesque configurations for sure. So this one can be reliably identified without microscopy because it uniquely has this bluish conidia. And pair that with these sterile white apices, there are not a lot of Xylarias that have blue conidia. So if you see somebody saying like, oh, that's Xylaria polymorpha and it's got like white conidia or something, you can automatically be like, no, Xylaria polymorpha is... Fenner taught me that Xylaria polymorpha is one of the few ones that has bluish conidia, so that can't be right. And the teleomorph's actually pretty easy to identify as well because it retains those grotesque configurations. It's just going to be a big mass of nasty Xylaria. And they get super rugose. They get very wrinkly as they age. You can see these pictures are by a fellow BMS member, Amos Zoller. So yeah, that's Xylaria polymorpha, the dead man's fingers. It's got a pretty cosmopolitan distribution as well. It occurs in Europe. It occurs all throughout the Midwest and United States and East Coast. It's all over the place. So this one seems to be one of the more common ones in my locale, at least. So I thought I'd cover it next. This is Xylaria cornudame, and this is a very textbook specimen. You won't always see them looking so perfect, but this pretty much has all the features that you would see on Xylaria cornudame. This one grows from really, really well decayed wood, sometimes even buried wood, like off the side from a really rotted log. It likes really mossy wood too. And this one can be reliably identified without a microscope too, if you know what to look for. It's got this white conidial coat, and this one's really on there, but even whenever it gets really, really mature, there will be traces of this white conidial coat the whole time. It's very, very uncommon to find this with no conidia on it, even whenever it's totally sexually reproductive, 100% teleomorph, it's going to retain that coat. And the epithet cornudame means like a deer's horn, so it likes to branch out. It doesn't always, but it's often very branching. And I got this close-up picture here so you could see those parathecia starting to kind of poke through the conidial coat. It gives it this really nice, like cookies and cream kind of vibe. I like it. I think I got a picture of the anamorph here too. Yeah. So the anamorph, same, it grows like, for some reason I find it off to the side of really rotted logs. It'll be growing from like buried wood or really nutrient dense soil. And you can see that's got this really chalky, thick layer of conidia right here. And right down here, you can barely start to see these juvenile parathecia just poking through. And something unique about the anamorph here is it always has these blunted apices. And if you look really closely, you can tell this is consistent. And every time I found the anamorph, the apices are like a very slightly off-white, and there's actually no conidia on them. They're blunted and they're sterile. So this is Xylaria longiana, and I slapped Sensu Rogers on there because I personally think this taxon's kind of dubious. It was described from Texas on oak bark. And I find it on oak bark here from time to time, but I mostly find it on decorticate oak, which is weird. And it's got for it to be described from Texas, it is kind of all over the place on the East Coast. I wonder if this might be a species group. So, this is a pretty cool one because it's got whenever it matures into teleomorphs here, it sort of leaves like this snakeskin-like pattern on the stroma. It's a pretty small species. It gets maybe 5 to 10 centimeters tops. It can grow decently long stipe or no stipe at all. This is one that you should probably do some microscopy because it's super variable. But this is a pretty textbook specimen right here. Except for this guy. I'm not sure what's going on with him. And here's the anamorph of Xylaria longiana. As you can see, not a whole lot of defining features to work with there. And then you couldn't hardly really call it anything but Xylaria with that. But here, you got more teleomorphs, and you can see that snake skin pattern. That's consistent. You'll always find that on the teleomorphs. Pretty small species as well. So here's one that I've actually never found, but a lot of people have, so I know it's around here. I also slapped Sensu Rogers on this one because Xylaria multiplex was described from Mexico, and we're a pretty good ways away from Mexico, so the pretty good reason to have some doubt there. But I mean, it looks and behaves just like the Xylaria multiplex from Mexico. So until like a comprehensive modern phylogenetic study's done, I don't see any problem with continuing to use that name. This is also a picture from Amos Soller. And this one, it's pretty similar to Longiana, but it's much larger, and it grows in these super cespitose clusters, which is pretty unique. It especially likes the bark of oak as well, just like Longiana, but much bigger and much more cespitose growth. Really like to find it one day. Here's a really cool one. It's the closest relative to Xylaria hypoxylon that we have on the East Coast. It's Xylaria vasconica. It was actually described from Europe as well. We just happened to get it here. And this one can also be reliably identified if you know what to look for. And what to look for this one is it is the only East Coast species that, as the parathecia develop, if you look closely, you can see it causes these horizontal constrictions on the stroma. These clusters of parathecia just kind of all develop, and it makes these, like right here, these horizontal constrictions. It's pretty similar to Cornudame, especially ecologically as well. Like, it likes really well-decayed wood. It especially likes mossy, well-decayed wood. But it doesn't retain that conidial coat like Cornudame does, and it makes these horizontal constrictions. And I think it broke off here, but the apex is always slightly attenuated, a little bit pinched. So this is the closest you're going to get to Xylaria hypoxylon on the East Coast, which we don't have. We have that on the West Coast, but we don't have Xylaria hypoxylon in the strictest sense on the East Coast. So here's the anamorph to Xylaria vasconica, and you can kind of see how it might have an affinity to Xylaria hypoxylon here. The anamorphs are practically indistinguishable. So if you didn't know that Xylaria hypoxylon doesn't occur here and you found this and you called it that, really couldn't blame you. They look exactly the same. So here's one that I'd really like to get better pictures of, but and one that I would highly recommend you do microscopy to arrive at the identification is Xylaria corniformis, also described from Europe, but occurs here. This picture is from Django Groupmeers, and not a too terribly too much features to work with for this one. So that's why I recommend microscopy. See, it can be super flattened and highly branched, or it can be like this one, and it's just a single stroma, pretty uniformly clavate. Like, there's no rhyme or reason to what Xylaria corniformis does, but it can be identified with microscopy pretty easily. It has uniquely small spores. And this is actually you won't hear me say this often, but Xylaria corniformis is actually easier to identify as an anamorph. And that is because it, as well as Xylaria polymorpha, is one of the few Xylaria species that has that bluish conidia. Again, it's kind of hard to see here, but it's got the same kind of color as Xylaria polymorpha, that smoky kind of navy blue. And it's also got these super tomentose base right here, like a very bulky tomentose base, almost like electric blue there. So even though it has blue conidia like Xylaria polymorpha, if you saw them growing right next to each other, you'd never confuse them because like these are not grotesque configurations. They're very uniformly clavate, and they've got this robust tomentose base that that the stroma arise from. This one likes pretty well decayed hardwood, like a lot of others. I've only ever found it once or twice. I didn't really get great pictures of it, so I'd like to find it again. Here's Xylaria tentaculata. We were just talking about this one a little bit earlier, and I wanted to use this picture from Maricel Patino in particular because it shows something a little unique about this. Right here, you can see, it's a little hard to see, but it's super rough, right where all these projections are coming out from. It sort of flattens out and gets roughened with parathecia. So that is where the sexual cells, the sexually reproductive cells are formed in the parathecia right there. They actually are not formed on the sparkler parts, if you will. They're formed more like where those protrude from. And you'll find this one on super, super decayed hardwood, like to the point where it's practically just soil. You'll find it on leaf litter. I've found it on conifers. Like, it's not super picky. So here's one from Alicia Milliken. This is Xylaria mali. So here's another one where I get to use the super fun terminology, grotesque configurations, because, buddy, it makes those, no doubt. The epithet Mali is in reference to its affinity to apple trees and crab apple trees in the genus Malus, and it has a high affinity for Malus. It grows on the roots of apple trees mostly, but it can be opportunistic and hop to other hardwood roots. Like I think it's been found on oak. I think I identified one growing on oak roots even. So it's not super picky, but it does seem to like Malus a lot. So it's uniquely got this sort of flattened stromata, flattened, grotesque, sesspitose. It's got this chalky white conidia, and the apices of the stroma are a different color than the conidia covered parts as well. It's sort of like an off-white apex and that's pretty consistent. So if you're ever around an apple orchard or so and you see some grotesque configurations of Xylaria popping out from the ground, probably Xylaria mali. Here's one that I'd really like to find, but I'm not even sure it occurs in Kentucky. It might be a more boreal species. I want to say it was described from Michigan. There's a few observations in like northern Ohio as well. And it's described as being the sibling species to the European Xylaria digitata. And this one is Xylaria acuta. It's named acuta because you can see the apex is pretty consistently like attenuated and pinched. It can grow from pretty long buried stipes. All I really know is it likes hardwood and it seems to maybe even just like colder climates, but I'll be on the lookout for it. I'm not sure if it occurs in Kentucky, but I'm gonna keep looking. This is from Igor can't remember his last name on iNaturalist, but he let me use a handful of his pictures because he does really good work on Xylaria as well. Another one I'd really like to find in another picture from Igor. I actually just added this slide today because I forgot that he gave me permission to use his pictures. So this is Xylaria frustulosa. It's obviously very unique for a Xylaria because it's not really upright. And I know you heard me say, like, hey, Fenner, Xylaria is characterized as being upright stromata. What's up with that? Well, this group of Xylarias are called penzigoid Xylarias in reference to the genus, or maybe outdated genus, might not be a valid genus anymore, Penzigia, which is basically just flat Xylarias. But a little bit of the stroma kind of digs down into the wood, so it's not truly flat. It's sort of erumpent. But super unique species. It seems to grow on decorticate hardwood. This collection was on decorticate dead but still standing white oak from I think Igor's in New York. Don't quote me on that part though. But I'm not sure if it occurs in Kentucky either, but I'd really love to find it one day. It might be a more northern species as well, but it's super unique for a Xylaria. All right, this one is Xylaria longipes. It is very, very common. It seems to prefer beech, maple, and occasionally sycamore. And it's got the blunted apex. It grows from a sterile stipe, and usually it kind of has like a kind of widened base. It's kind of hard to see there though. And something unique about this, well, I guess not totally unique because the next few we cover also do this, but this the ectostromal surface cracks into these angular scales right here. You can see it really well. And that's as the stromata matures, it just begins to fissure sort of into these scaly patterns. And this one absolutely needs microscopy to identify it. Like I've tried to come up with ways, but anytime I think I've got something that works, I end up proving myself wrong. You just need microscopy. What's unique about this one, microscopically speaking, is it's one of the few Xylarias that's got this sigmoid germ slit. And sigmoid just means spiraling. So you see it's not a straight germ slit, it just kind of spirals right there. And you can hardly see it there too. And it's got sort of chestnut brown colored on spores instead of just like a stark black. You'll find it, like I said, you'll find it on maple, beech, sycamore, but pretty much always decorticate. And you'll see it spalts the wood as well. Here's the anamorphs, and this right here technically isn't an anamorph. The literature would call this Ascadrus clava, but I call it Ascadrus stroma. I just think it illustrates the picture a little better. So these right here, you can call them anamorphs because they're covered in conidia, but this one in the middle is totally sterile. It's just there to eventually give rise to asci, which is what Ascadrus literally means. So, and again, you see it on that decorticate. I think it's maple here, decorticate and spalted maple. And that's Xylaria longipes. So, this next group that we're going to cover basically looks exactly like Xylaria longipes. That's why I say you need microscopy for this. However, the anamorph of Xylaria flabelliformis, pretty easy to identify. But this right here is a mature stroma placed right in front of an anamorph. So if you didn't have the anamorph there and you just saw this clavate blackened stroma, you'd have no way of telling if it was longipes, flabelliformis, or even the next two that we're going to cover here. But the next two, following this belong to this group. It's known in literature as the cubensis aggregate clade. It's a pretty cohesive phylogenetic clade that I don't even think Xylaria longipes belongs to, but these next ones do. They call it the cubensis aggregate clade. I just call it the cubensis group. It's a lot easier to say. But yeah, they all have this clavate stroma, and they all have really wacky anamorphs, which is super cool. Flabelliformis has a really cool anamorph. It sort of looks like chewed up bubblegum, and I'm sure many of you have probably seen this one. Actually, I'm not sure if it's in Wisconsin, but it wouldn't surprise me because it's literally everywhere. Yeah. So here's again with the Ascadrus stroma that will eventually give rise to Parathecia and Asci. And I've actually tried keeping like a log of this in my backyard before, so I can like kind of incubate it in a sense, just watch it throughout the season. And as the Ascadrus stroma mature, the animorphs seem to just go away. Like the animorphs are not there to develop into the mature teleomorphs. That's what these are here for, which I thought was really weird. It's the only species I've had the opportunity to do that, though. I've tried it with a few different ones, but like slugs and stuff always get the best of me. So here's another one belonging to the Xylaria cubensis group. This one is Xylariaceae. And again, it looks just like Xylaria longipes, looks just like the teleomorph to Xylaria flabelliformis. But this one has ever so slightly larger spores, longer spores than flabelliformis. So they can be identified. If the anamorphs aren't present, you can identify it by measuring spores. And again, just like flabelliformis and longipes, it has this fissured ectostromal surface that kind of crackles into these angular scales. That's a trait that's unique to longipes and the cubensis group. This one, you can't hardly see it because it hung out in my tackle box for a little too long, but you'll have that. And here is the wacky anamorph of Xylariaceae. It's sort of, you can kind of tell how it's got an affinity to flabelliformis, how it's got these like wrinkles and folds. It's not quite as dramatic as flabelliformis, but most importantly, what separates them is this cinereous conidia. It's very gray, smoky gray conidia. And it's not so chewed up bubblegum looking like the flabelliformis, but it does have a similar anamorph. And I've never found the anamorph, but a few people have reached out to me, and I know what the anamorph looks like, and this is definitely it. I'd really like to find it one time. It's one of the weird situations where I actually want to find an anamorph. I've got a lot of Xylariaceae collections, and like none of them are anamorphs. Oh yeah, this one was actually, go back. Okay. So now I forgot what I was going to say. Sorry, brain fog. Okay. So the last one in the Xylaria cubensis group is Xylaria maumii, which is a pretty interesting species. If I remember correctly, I think it was described in like the 1920s, and I don't think it's been collected ever so much since then. It was described from like Toledo, Ohio, right around the Maumee River, which is where it gets its epithet maumii. And it looks just like Xylaria longipes and flabelliformis and Ellisii, especially right here. This one's kind of a strange stroma. It almost looks like two fused together, but this is much more classic. It's got the ectostromal surface that shatters into those scales, and it's got the clavate blunted apex, and it grows from that widened base. So essentially, you definitely need to do microscopy if you ever find a Xylaria that looks anything like this. This one, all of the cubensis aggregate clade or the cubensis group have straight germ slits. Flabelliformis has the smallest. Ellisii has the middle range spores, and this one, Xylaria mammii, has the largest spores out of all of them, the longest ones. And they also have straight germ slits. And just like all the other members of the cubensis group, this one also has a super fun and wacky anamorph. So it's kind of hard to see here, but it is sort of like salmon pinkish, not quite as pink as Flabelliformis, but certainly not white. And like, I don't even really know how to describe that. Like that is just, it's sort of flattened. It's the conidia is densely on there. Like you can see this one's sort of more spathulate and flattened, which is pretty typical, growing right next to this Ascidurus stroma. But you can see where I placed this anamorph here. All that conidia just shed off like effortlessly. That's something I've noticed about this species. It really puts a thick layer of conidia on the anamorphs. All right, so this one is, I have concluded anyway, that it is doesn't have a name, doesn't have a real name yet. But it can be identified without microscopy really easily because it has a super unique ecology. It always grows right at the base of living oaks and beeches. So it's got here's the anamorph, just chalky white conidia, pretty sesspitose growth, blunted apex, all that. This one was at the base of a white oak, Quercus alba. I found it on scarlet oak and red oak as well, and also beech. It grows on beech as well. Here's the teleomorph. This one's growing at the base of a beech, sort of grows like right at the root collar almost. I would suspect it's probably a parasite, but there's literally no literature on it. Don't really know that. I've looked and looked to find a name that matches this one, and to no avail. So my conclusion, probably doesn't have a name. But the temporary code name that it's going by is Xylaria species Indiana 05. Super common too. I wouldn't be surprised if you guys had this one out there as well. It's I think I've seen observations of it in Alabama, North Carolina, Kentucky, Indiana, obviously Indiana 05. Like it's kind of all over the place. But just look around at the base of oaks and beeches. So this one has a super cool epithet. It's Xylaria finismundoensis. And this was described by Dr. Rue Vandegrift just a couple years ago, actually. It went a few years without having any other collection than the type collection. And proudly say I made the second collection. That's it right here. And finismundoensis translates to from the end of the world because it was described from the World's End Peninsula in Boston, Massachusetts. So I just think that's so cool, super creative from Dr. Rue to name it that, the Xylaria from the end of the world. This one is a super small species. I want to say it was like 4 to 5 millimeters in length. So like, and that's like the largest one right here. So small. You pretty much have to be looking at something else and then notice it to ever find it. But it grows on Decortica oak, it seems. So it's also has the sort of snakeskin pattern like Longiana does. It actually is kind of closely related to Longiana. It's in that Apiculata arbuscula group where it's just like smaller xylarias with the snakeskin conidia. But this one needs more documentation, so it would be really cool if people found it more, identified it more, because the distributions, there's not really a whole lot known about the distribution. So here's another really, really cool one that I have been looking and looking for since I found it so I can get better pictures of it. but I've been looking for 2 years now at the same spot that I found it, and it just hasn't come back. This is Xylaria morganii. It seems to be kind of rare, honestly. But it's got a couple of really unique features too that set it apart from other species of Xylaria. It's got these blunted apexes. It grows from, I found it on super, super well decayed hardwood right in the middle of this sort of stump. And it's huge. It's gigantic. It's like bigger than Xylaria polymorpha. You see this one's like 4 inches in length and almost an inch in diameter in other parts. Like that's really, really, really big for a Xylaria. And the features that are really unique about it is, so as you heard me mention that Xylaria typically have these globose or subglobose kind of spherical parathecia. This one has elongated parathecia, which I've never ever seen in any other species of Xylaria. And I apologize, the quality of these pictures are not fantastic because they were taken with my cell phone through a stereoscope, but it also has this white conidial kind of scales left over. But the elongated parathecia is super unique. So if you like think you found it in the field and you just kind of do a quick little cross section and you see the parathecia are not perfectly globose or subglobose, they're more elongated, you'd be like, this might be Xylaria morganii. Man, I'm about to make Fenner's day. Because I'd really like to have more collections of that. I have one, or I have 2, but they're from the same collection on the same day. So this next group of Xylarias are the fructiculus species, so known to occur on fruits and seeds. Excuse me. Okay, this one is Xylaria liquidambar or Xylaria liquidambaris, depending on who you ask. I think the current taxonomical situation with this is Xylaria liquidambaris, but I haven't updated that. And all of these can be really identified really easily without a microscope because they're substrate specific. So that's something that makes them fun. You don't have to work so hard on this group. And the teleomorph of this species isn't often shown, so I thought I'd show a picture of it as well. It's pretty unique. It's got this sterile apex here, and it also has a sigmoid germ slit on the spore. But one more thing about this species before I move to the next one is that it might actually not be a valid species. So Jack Rogers put out the name Xylaria liquidambar without really having conclusive data as to whether or not it's a synonym for Xylaria persicaria, because Xylarias occurring on sweet gum pods in classical literature were always called Xylaria persicaria, which is actually a Xylaria that's known to occur on peach pits. But I think, if I can remember correctly, like a lot of our peaches and stuff, we stopped growing peaches a long time ago, or so many of them, and stopped finding this Xylaria on peach pits, started finding it on sweet gum pods, and just called it persicaria until Jack Rogers was like, it's probably not that. So if you end up finding a Xylaria on peach pits, you might be able to deprecate this name. Or even you might confirm the late Jack Rogers hypothesis that they are unique species. So if you live near a peach orchard or know somebody with some peach trees, go check them out. So this one, super easy to identify. It grows on magnolia cones. It's Xylaria magnoliae. And unfortunately, I don't have any pictures of the teleomorph, but it's pretty much just like this one, how it's sort of got this fertile section. It's got a stipe, it's got a fertile section, and then it's got a sterile apex. That's sort of what the teleomorph for Xylaria magnoliae as well looks like, but these are just anamorphs. It grows on magnolia cones, and if you find a Xylaria on magnolia cone, it's this one. No need to whip out the microscope. So this one is Xylariaceae. I hear most people actually find this on the seed pods of hawthorn in the genus Crataegus, but there's quite a few hawthorns around where I live, and I've never found it on those. I've actually only ever found it on pignut hickory, which it also grows on. It grows on multiple species of hickory, but I always find it on buried pignut hickory shells, usually broken up into pieces. This one's pretty unique. A lot of observations of people finding like flies being attracted to it, and I've even heard people describe this one as like stinking really bad. I've never had the pleasure to smell a stinky Xylaria, but apparently this one can be. So, and it might even be there to attract flies. Might be some pretty unique research opportunity with this species. But and the how I was talking earlier about how the anamorphs and the conidia of Xylaria sort of have a dubious function. The paper is actually in reference to this species. See if I don't garble this. I've got brain fog. Bear with me. But it was something like the anamorphs come out of the ground right around the same time as the flowers bloom, and as the nut and the seed starts to develop, Jack Rogers in this paper, he would culture the endophytes from the inside of the nut, and they would find this species on the plate before the seed even hit the ground. So sort of seems to suggest there might be a sort of conidial pollination going on. I think that'd be really cool for somebody to follow up on that research. Do it for Jack. He did a lot of great work. So all of these species that I covered are actually pretty cladistically cohesive, like phylogenetically speaking. They belong to the Xylaria carpophila group, and that includes Xylaria carpophila itself. And for a long time, there was no records of this in the United States. It was only ever documented in Europe on European beach mast, but it's here. I found it on American beach mast. Here it is. It's Xylaria carpophila. Always buried. So if you live near beach trees, what it's going to look like is these tiny little Xylaria stroma coming straight out of the ground. But if you carefully excavate that, you'll see it's growing from beach mast. It's got a really cool teleomorph here. It's got the sort of snake skin. You can see, I especially like this right here because you can see these sort of like shredding open the stroma as the parathecia begin to develop. This one is Xylariaceae simplicissima. I threw CF in there because this is an anamorph. There's not really a whole lot I can do to comprehensively identify this. I think it was put into a tube to get sequenced and stuff, but this was only pretty recently moved into the proper genus Xylaria. It used to be Rhizomorpha simplicissima, and it like gets misidentified as different Xylaria that I can't remember the name of. It's the one that grows from leaf litter really often and petioles. Oh, it's gonna bother me later. But basically, it was misidentified for a long time. Filiformis, Xylaria filiformis. Yuming Zhu, world expert on the genus Xylaria, looked at over 800 type specimens. He's absolute legend. Went through a whole lot of Xylaria filiformis collections from the United States and found that like literally all of them were Xylaria simplicissima. So Xylaria filiformis might be here, but doesn't seem super likely. Seems way more likely that we have simplicissima. This was found by Michael Weiss at Learning Man. It grows from leaf litter, petioles, and it will also grow from herbaceous stems occasionally. So yeah, I think that's it. That's all for me. This is all my references. This is what I used to come to the identification of pretty much all of those. If you want any of these papers, you find yourself finding them behind a paywall or something, just reach out to me, get you the paper.
Awesome. Thank you, Fenner, for your amazing talk. We do have a number of questions
Awesome.
in
Let's
the
get into them.
chat. So I'm gonna have to scroll way back up to
I
find the
had
first one.
copied some of them. You can check if I miss any, but the first one I saw was from Lauren, Ray, and Jack. Actually, I think Jack wrote it, Jack Johnson.
Yep.
He answered it, I'm pretty sure.
Oh, okay. You had said Xylaria multiplex turned up in Hawaii as well.
Ooh, cool.
There are a handful of species known to occur in Central, South America, and Hawaii, per Rogers and a handful of ITS results. Oh, and then your question was, oh, sorry, this is Todd's question. So Todd Osmondson asks, do the tentacles on Xylaria tentaculata produce conidia?
Yes, they sure do. But they do not produce parathecia, only conidia. The parathecia get produced right below where the projections come out.
And Walker had asked, can you talk about toxicity in Xylariaceae? I've only heard rumors about polymorpha being super toxic. Not a concern cause like Xylaria is the last thing anyone would try to eat. But you say that, Walker, but I've heard of people eating them in Europe, I think.
I've also heard of it, yeah. I've heard of people putting Xylaria polymorph on steak tartare, which is super weird to me, but whatever. So I don't know
what the
an
hell?
awful lot about this. Yeah, but
Who's
I
eating
was
these?
just talking about it the other day, so it's kind of fresh on my mind, luckily. There was a paper that came out from some Indian studiers in 2018, I think, that found super small amounts of like amatoxins and phallotoxins in 5 different species of Xylaria, but the methodology wasn't made super clear, and it's like seems like pretty infinitesimally small amounts. Probably just needs another study done. I personally don't recommend eating them though, because they're a hotspot for novel and unique chemistry. I mean, you don't really know what you're eating. So I recommend not eating them.
So other people can unmute themselves and ask, but I'll just ask the last couple. So we did have, although I think Jack did say that he thought that you maybe already answered it, he had asked, how do you distinguish Xylaria longeata telomorphs macroscopically from Xylaria cornudame? Both have the white coating.
Okay, so Longiana is quite a bit smaller, and instead of like just retaining like a rough kind of white conidial coat, it has, like I said, that snakeskin sort of pattern, whereas Cornudame is quite a bit larger. It's very rarely grows directly from like decorticate or oak bark. It's usually on really well decayed wood, but yeah, the conidial coat's a pretty dead giveaway as well as the stromal size.
Thanks. Yeah, you got a good answer in the presentation. I was quick on the gun to ask that one, but that helps more.
Right on. Glad I could help.
In the chat, Lisa Wallner shared some of her Xylaria photos. One of them I thought was Xylaria cornudame, and the rest of them I didn't have a statement on.
That first one I would call Cornudame, yeah. The second one, oh, that's a fun one, needs microscopy. It's Longipes or cubensis group, yeah. You can tell it's that by the blunted apices and the, like I said, like too many times in that presentation, but the ectostromal surface fissuring into angular scales. That's a trait that's unique to those. And then let's see the third one, also Cornudame. Yeah, super common.
Cornudame is just a really nice species epithet. I just
like
like saying it.
Yeah, Cornudame, like a deer's horn.
I had another question I'd put down for later. How do you think these anamorphs in the cubensis group turn into those nice clavate shapes? Does the mycelium just abandon the anamorph Or I know you talked about the one in your backyard, but
Yeah, like like with Flabelliformis. I honestly think that there's like a lot of cases where the Animorph doesn't stick around. It's just like Animorphs next to Ascydrostroma. Eventually the Animorphs bug off and just fall away, do whatever, and then the Ascydrostroma stick around to become Teleomorphs. That might be the case for the other ones. I can't really conclusively say that. I haven't done any backyard experiments with them yet. I've tried though. Like I said, those darn slugs. I just love it.
I've always been curious about the germ slit feature because it's like such a hallmark of them. Is there do you know of anything that like, it's kind of a weird question, but like what does it do for the spore? Do they have any speculation about it?
It really might just be where germinates mycelium from, as far as I know. About like a germination pore or slit would be, yeah. I'm pretty sure that's the only function it serves, other than being a useful taxonomic trait for us.
yeah, those sigmoid slits looked really, really pleasing to the eye.
I know, right? Yeah, those are my favorite for sure. There's not a lot of Xylarias that do that either. It's Xylaria longipes, Xylaria liquidambarus, and I think even Xylaria acuta can be slightly sigmoid.
I was taking notes for later, and I did sketch out the sigmoid. I wanted to remember that.
Yeah, it's a fun word too, sigmoid.
Yeah.
Spiraling.
I have a microscopy question.
Yeah.
So how when you're ready to do the dirty and you're gonna chop it up and start getting spores on the slide, how do
Right.
you pick what part of the fruit body or what part of the collection to look for ascospores? Because it seems kind of like a gamble.
Yeah, yeah. Okay. So that's a really good question. Let me share my screen again, if you don't mind. So
I've made that gamble a
basically,
lot.
like you're asking like which part of the fruit body do you take from whenever you're trying to look for spores and stuff?
Yeah, like what tips do you have to avoid just like excavating empty perithecia that don't have
Okay.
ascospores in them anymore?
Yeah. So I've had that issue a few times actually, where you'll like cut. Actually, the reason why I do cross sections in the field isn't necessarily to get like swaggy pictures, but it's actually to see like if the perithecia are darkened and carbonaceous. Because if they aren't, if they're like really pale perithecia or something, there's no ascospores in it. But let's see. If you see right here, I kind of cut off just like that first layer of the ectostromal surface. And then if you just take like a really thin section of a perithecium that's like really blackened like this one, you'll find all the ascospores you could need. And if that's whenever I was first starting out with doing microscopy on Xylarias, I wasn't super good at getting thin sections of perithecia because let's face it, microscopy's hard. I used to just take a little needle and like scoop out some of the gunk in the perithecia and plate that up, and it works just fine. Sometimes I'm lazy and I still do that because it literally works just the same.
Well, that's what I've done in the past too, but it helps to know you can make the section. I don't know. Just
Yeah, I mean
destroy a
it's
fruit
kind of
body
cool to
with-
get like a picture of the section under like 10x itself, but like you don't have to. As
Yeah.
long as you're looking at spores, you'll probably be able to identify it.
There's a student at UWL who's doing scanning electron microscopy on Xylaria a lot right now, and
That's awesome.
if this talk's recorded, we will make sure CC can listen. But thanks for the tips. I'll
Yes.
distribute them.
And thank you for
Oh, my pleasure.
There's one more question real quick in the chat. Sarah Stokes asks, if there was one thing you would want amateur mycophiles to know and remember about Xylaria, what would it be? Also, she said she might have missed this from the beginning, but what got you so interested in this in particular?
Okay, those are good questions. I'll start with the last one. What got me interested? I kind of just noticed that nobody ever identified Xylaria species, and I was like, man, it'd be pretty cool if I could do that. So I went down the rabbit hole and figured out what it took to do exactly that. And I ended up getting kind of adept at microscopy and then getting really into it. And then I would end up finding these species that hadn't been found since like 1920. And I was like, I felt like the mayor of this town. So I really stuck to it, I guess. Super cool genus. And one point that I'd really like to drive home is like, don't let the need for microscopy kind of like deter you from studying the genus, because the biodiversity in Xylaria is mind-boggling, and it needs a lot of help taxonomically speaking. So it'd be just really nice to get people interested, get them the tools they need to identify them, understand how to identify them, start documenting them more, and really kind of fix this issue. Yeah. They're a slept-on genus, and there's too many names.
Yeah. Max Reinhardt asks, from a more beginner perspective, can you talk about the development between the telomorph and anamorph stages?
Right, okay. So early spring or late spring, early summer, the anamorphs start popping out. Was the question like what the differences are between them, kinda? Sorry,
I
I've
think
got brain
like
fog.
more of a like stages of like development, like what happens between the two is kind of what he's asking.
Okay, yeah. So as an anamorph begins to mature, eventually that conidial coat will just kind of slough off, and eventually it will start to develop these parathecia inside the stromata. So instead of having these external spores, it'll be internally born, and those will start to develop. A lot of the times you'll catch them like right halfway. I think I have a picture I could show you as well. There's a point in between anamorph and teleomorph where it's just completely sterile, is basically what I'm trying to say. So it'll be anamorph, and then it's going to be sterile as it develops into a teleomorph, and then the parathecia will mature, and then it will be a teleomorph. So all of that roughly takes about 4 or 5 months, yeah.
Just if anyone missed it too, like as you know, we're going over these terms like conidial, those are asexual spores versus in the anamorph versus in the teleomorph, they're sexual. So they're genetically distinct versus in the conidial stage, they're all identical genetically.
Mm-hmm.
Not for you, Fenner, you know, for everyone else.
Yeah, you're fine. A term I hear some people using is I actually really like it, mitosporic and myosporic. I get tired of saying anamorph and teleomorph, so I'm thinking I might start saying those.
That's cool.
Switch it up a little bit.
Hit me with those again.
What, mitosporic and myosporic, like mitosis and meiosis? Like your spores are either gonna have the full genome or not.
I'm gonna write that down. Write that down.
Write that down.
Any other questions for Fenner?
Thank you so much,
No?
Fenner, for doing this.
Yeah.
That was
Yeah.
an excellent
Pleasure.
lecture.
Excellent.
I hope I
Um,
delivered, because I definitely was feeling the brain fog.
absolutely. You did
You
great. And
delivered.
I couldn't tell you were sick at all. So that was amazing.